Find out about the little worm that's having a big impact on farming. Hi, my name is Grace Farmray and I'm a third year biochemistry student. I'm currently doing a bioinformatics project all about Hymunchus contortus. So I'm guessing that many of you have never heard about this organism before, but my project is all about Hymunchus contortus, a parasitic roundworm that infects livestock such as sheep and goats. Even though it is very tiny, 10 to 30 millimetres in fact, it can have big consequences. Infected animals develop anemia, swollen tissues, and in some cases it can lead to death. As you can imagine, this has a detrimental effect on livestock production due to losses in milk and meat production, as well as additional treatment costs. So, one question is, how does the infection spread? Well, Hymunchus contortus is transmitted orally through the livestock grazing on pastures which have been contaminated with eggs, and these eggs are excreted along with the infected animal's faeces. The warmth and the moisture of the soil is a perfect environment for the eggs to hatch into larvae. Other animals in the field ingest these larvae whilst grazing. Once inside, the larvae develop into adult worms which feed off blood capillaries inside the stomach. So, why is this roundworm worth doing my project on? Well, the majority of drugs used to treat these infected animals are now ineffective, as Hymunchus contortus has developed resistance to these drugs. So the more that we understand about the molecular pathways that govern this organism, the more likely it is we can find new potential drug targets. So this makes Hymunchus contortus an important organism to study. My project involved looking at the protein kinases of Hymunchus contortus to see if there were any expansions or reductions in the protein kinase families. This was to try to identify protein kinases that were specific to the parasitic life of Hymunchus contortus. This involved using a number of computer programs. First, UniPro to align the protein kinases so I could see if they contained the three essential motifs. If they contained these motifs, I was able to class the protein kinases as active. The next step was to use PFAM to see if the sequences contained a protein kinase domain and if it was truncated or full. Finally, I used UniPro to conduct a blast search, seeing if the protein kinase sequences had any sequence homology with sequences from other organisms. Using all this information, I was able to highlight particular families and sequences which seemed to be specific to Hymunchus contortus. I really enjoyed working on this project and I hope you found this video interesting and informative. Thank you for watching.